Hello and welcome to Enago's webinar, organized exclusively for the researchers of Prince of Sonla University and the partnering universities. This session will help researchers prepare an error-free manuscript for submission in top-ranking international journals. Before we begin, I would like to mention that you can send us your questions using the Questions tab in the control panel, and our expert speaker would be happy to answer all your queries at the end of the webinar. Today, we have with us expert speaker Dr. Despina Sanoudou. Dr. Sanoudou holds a PhD degree in molecular cytogenetics from the University of Cambridge and currently works as an associate professor at the Medical School of University of Athens. Dr. Sanoudou is a renowned medical researcher and a published author with over 178 publications, 5,000 plus citations, and over 20 grants to her credit. She has also been associated with Harvard Medical School as a postdoctoral fellow and an instructor. In today's session, she will explain in detail the standard structure of a medical research paper and how to write each section comprehensively. We extend a warm welcome to her and thank her for joining us today. Before I hand over the mic to Dr. Sanoudou, I would like to give you all a brief introduction about Enago. We are the world's leading author services provider, and our author first, quality first approach ensures that our authors receive only the best quality output and excellent user experience at all times. This unrelenting focus on authors' needs means that we never compromise on our quality systems and processes at any cost. All our services are designed keeping in mind authors' needs. Enago Academy is our author training platform that educates authors through several resources, including webinars as well as live workshop sessions. We also have an e-learning platform for each researchers called Enago Learn, along with live one-on-one consultation platform called Enago Consult. Enago offers a world-class service to researchers at different stages of their research career, such as two-editor system English language editing services, academic and non-academic translation services, manuscript editing services, and publication support services. The Enago promise guarantees that a manuscript edited by us will never be rejected based on poor language. On a rare occasion, if the rejection occurs, we will take full responsibility for re-editing the paper for submission at no additional cost. Enago's comprehensive after-sales service will support you at every step of the way until your manuscript is published. They are designed to take care of every need from time you receive our main service until you are published. Enago's research communication service are designed to help you effectively present your research data. In case you are interested in any of these services, feel free to visit the dedicated portal for the researchers of Prince of Sonla University, Faculty of Medicine, and get discounts at preferential rates as a part of the partnership. For other participating universities, please reach out to us via the email address displayed in the chat box to learn more about the institutional partnership opportunities and benefits. Without any further delay, I will hand over the mic to Dr. Sanoudou.
Good day, ladies and gentlemen. It is a pleasure to be joining you today for this presentation, and I thank Enago for the invitation. Today, our agenda will include an overview of medical publishing, conceptualizing your research, useful tips, advice on best communicating your research findings, medical publication and research ethics, a very important aspect, and how to prepare and evaluate case reports. Let's start with an overview of medical publishing. Why is it important to publish your research findings? Well, first of all, it is an integral part of the career progression process. We are evaluated regularly in the academic track for our publications, the quality first and foremost, but also the quantity, how much work are we producing, how many new scientific findings, how well are we communicating it to the international scientific community. It's a part of personal development, because with every new project and every new publication, we are learning something new skills, new knowledge, new ways to communicate our findings. It's also a means to promote our personal networking and therefore expand our network of collaborators with indirect advantages, significant advantages for the first point, our career progression. It's a very important part of our contribution to the clinical and public health. Unless we publish our findings, the world will never know. They will be hidden in a drawer somewhere in our office or in our lab. So in order to put them into good use for the general public, we need to publish them in a timely, accurate, and appropriate fashion. It's a major contribution to science, because if we publish our findings, then other scientists around the world will be able to use them in order to build their research projects, fit the different pieces of the puzzle better together in order to answer more scientific questions. So unless we communicate, we publish our results, we're not helping the scientific community or the clinical setting. So the true value of our research output, clinical or basic research output, is publication and communication to others. And a fifth very important point is the channelization of time and resources used for practice. Let's look at some useful facts from publishing in medicine. So, non-publication of research is unfortunately a common occurrence. What do I mean by that? A lot of the work that is produced, a lot of the research that is performed never gets to be published. Why? Sometimes because of lack of time or low priority. The people who are performing this research, possibly due to all their other commitments, do not have enough time to put these findings together into a publication. Non-submission is a more general term, but refers to possibly preparation of the work in the form of a manuscript, or putting together the results in the form of notes, but never actually submitting it for publication for various reasons. This is unfortunately a very common occurrence. It's up to 85% median. And then there are studies that are incomplete or still ongoing, and sometimes they're left incomplete. For example, if a student who's performing the project leaves the lab and there's no one there to continue it, or other projects that are still ongoing. Another important fact is the lack of awareness. Out of the 50% resident doctors that had participated in research other than a dissertation project, appeared not to be aware of the possibilities for publication. 28% had made scientific presentations, but only 4% had actual publications. So these percentages are very low. The rejection rate is another important reason for non-publication of new discoveries. According to a study by Hall and Wilcoxon, the conclusion was that 60% of the published papers have been rejected at least once. So you can imagine how, and we will see in more detail, how big the rejection rates can be and what implications this has. The rejection rates for various top-tier journals can vary between 80 and 85%, while in some other top-tier journals it can reach up to 90 or 95%. These are very, very high numbers and they appear scary at first. However, with careful, appropriate preparation, one can definitely increase their success rate, their personal success rate when it comes to publishing. And this is what our presentation today is really all about. Let's look at some more publishing facts from the Imago source of information. This is a graphical representation of the publications per country over the period of 1996 and 2020. The size of each circle represents is related to the number of publications, and you can see how can these vary from country to country. You can also see the number of citations per document as an average. So Thailand in specific, you will see that over that period of time had 69,636 publications, and each document received on average 17.64 citations. The H index, if you look at the country as a whole, was 288. Now, if we look into more details, you will see the exact numbers again from the Imago Journal and Country Rank source. You will see the actual number of publications, citable documents, citations, and self-citations, also important to bear in mind, as well as the H index for each country as a country. And Thailand ranks 39th, very high, with, as I said, 69,000 approximately publications and an H index of 288. More details on country or institute-based statistics can be found on various web resources, and they're helpful to have an overview of how each country or each institute is performing. Now let's look into a little more information about rejection and acceptance rates in medicine and specific in medical journals. So, for example, the New England Journal of Medicine publishes only 5% of the 5,000 research submissions they receive every year. So you can see how they're actually receiving a very, very large number of manuscripts, and they have an extremely low acceptance rate. So a very competitive journal to get published in. JAMA Internal Medicine receives approximately 3,000 submissions per year and has a 13% acceptance rate. Needless to say that as we go on to lower and lower impact factors, the acceptance rate is increasing because the number of submissions per year is decreasing. So Okiki et al., another useful piece of information, found that in 5 years, 76% of manuscripts that were rejected by the Journal of Bone and Joint Surgery got published in other journals. So there's some important take-home messages from this slide. First of all, high-tier journals are extremely competitive. Needless to say, that's because it's so prestigious to have a publication in a journal such as these. 2, although this is extremely competitive, it's also a challenge and an incentive to keep improving as a scientist, as a writer, as someone who wants to publish their work, in order to be able to reach as high an impact factor as possible, and as prestigious a journal, I should say, because impact factors is not the only parameter, but as prestigious a journal as possible. And another take-home message from this slide is that even if your manuscript does get rejected, There are many ways to improve it. There's many things you can learn from the process and target your next submission of the same or other manuscripts to a different journal, possibly better suited for your work. Okay, let's change pages now and move into some useful tips on conceptualizing your research study and getting going. The first extremely important starting point is formulating a research question. What will your work be focused on? This is the first thing to decide when you're starting a project. You need to find your focus, and in order to do this, you need to observe. First of all, decide on the field that you want to work in. 2, read as much literature as you can, talk with expert scientists in the field, and identify those important scientific questions, medical, biomedical, basic research questions, whatever it is that you are most excited about. Identify the open questions, the hot topics, if you want, in your field, and then read some more in order to determine who is working on what and using what approaches. Carefully analyze those gaps in scientific knowledge that are there, and through your detailed, careful literature review, determine where there is room for you to build your project in order to be able to contribute something truly novel on an area that there's actual need and there's heightened interest at this period of time. You may need to go through multiple rounds of evaluating the literature, talking with experts, and considering and reconsidering your exact scientific question. It's extremely important, all of these points, because you don't want to start a project, to spend all this effort, time, and money only to find out 2, 3, 4 years down the line that what you just completed has already been published, is already publicly available, and you've been working towards something that is no longer really publishable or not publishable in the level of journal that you would have wished. So carefully question yourself, be yourself's toughest critic, and reassess and discuss with people you trust who are also highly knowledgeable in this field in order to fine-tune and determine the research project, the research question you will be working on. From there on, there is a series of crucial steps to take and considerations to have in mind. So in this slide, I've tried to summarize the key components of the research process. Each one of them could be an entire talk. However, I still feel there is need to point them out here and provide you some basic information on each one of them. Once you've decided your research question, you need to plan and strategize. What approaches will you use in order to answer this scientific question? What experiments will be needed? What clinical cohort or cohorts will be needed? What will be the inclusion and exclusion criteria? How long will the project take? In what time periods do you need to complete each part of the project? How well will the different experimental components answer the different sub-questions of your research project? What controls will you need to include in your study? Number 2, acquire all resources and collaborators. Often, the scientific questions we're trying to answer require the collaboration of people from multiple disciplines or people, experts with different technical skills, different know-how, different scientific knowledge. It's important to decide that, to appreciate that right from the beginning and identify those people who would be well-suited collaborators in order to perform this project, to complete this project efficiently, effectively, and fast. You need to know right from the start what resources you will be needing. Do you have the necessary funding? How can you get the necessary funding if you don't have it? What equipment will you be needing? Will you be needing to pay salaries? What consumables will you be needing? How much do they cost? When will you need to purchase them? What's their expiration date? How much will you need of them? All these are technical pieces of information that are, however, crucial when it comes to building the foundations of your project. Number 3 is the point where we comes to conducting the study, and this needs to be done meticulously, and I would write this with capital letters. You need to be very clear as to what it is that you're doing and how you're doing it. You need to have all the controls in place, ensure that everything is recorded in detail, every experiment, every day, even if it's the same protocol over and over again. Every day you have to write down, and your colleagues and the members of your team need to carefully keep track of everything that's being done. Be aware of any deviations of the results from what you would expect, reasons for deviations, any contamination that may take place. Be very careful so that there are no sample mix-ups and that you have ways to check and confirm there were no sample mix-ups, etc. There's really so much to be said about each one of the points in this slide. Number 4, compare and contrast your findings to existing knowledge. To some extent, some of the information you will be discovering, some of the results that will be coming out of your data will be along the lines of work that has been performed previously. Some parts of it, make sure that you compare and contrast regularly your findings to the literature. What it is that other people found, what is it that you're finding? Are these in line? Are they consistent? Are they pointing towards the same direction? Or are you seeing major deviations? Are you observing something totally different? And if yes, why? What is the reason for this difference? Is it something that you're doing better? Is it a different protocol? Is it just a biological difference that's observed in the 2 different biological systems that you've potentially been using? Or could there be some problem, something that went wrong, either in their side or your side? This is why it's extremely important to regularly compare and contrast your findings to the literature, not necessarily wait until the end. Because if for any reason what you are finding different is due to a problem, something going wrong in your experiments, then you will be sensitized early enough in order to look into it, correct it, and continue effectively with your project. Once you've gone through these 4 steps, which can take a fairly long time in some cases, then you're reaching the point where you have all your data, all the information, and you're ready to communicate your findings and conclusions to the scientific community and possibly the general public. So how do we do that? Compiling your research. First of all, you need to make connections between existing literature and your research. So you need to, again, carefully read, be aware of everything that's out there, and discuss this in your manuscript, or make notes how you will be discussing this in your manuscript. Group your ideas and data under different headings. So start organizing the information you want to present. Try to answer these questions ahead of starting your write-up. What is the aim of my research? What can I demonstrate based on these data? So make sure that the scientific question that you're trying to answer in the manuscript, not your project anymore, but the specific manuscript, is very clear, and that your data can address this question. What I often do before I start writing a manuscript is I write on a piece of paper the question that I want my manuscript to be addressing, the scientific question. And literally, I tape this piece of paper on my computer screen, on the side of my computer screen, so that every minute throughout the time that I am putting my manuscript together, I can look at this question and make sure that everything I write is aligned with the question I'm trying to answer. Because often you have a lot more data than what you will be including in your manuscript. What you're including needs to fit into a nice story. Organize your data while maintaining the flow of your article. This is extremely important as well. We can't just dump all our data in there and wait for the reader to make sense out of it. No, you need to carefully decide, again, what is needed from your data to answer the scientific question that this manuscript is addressing, and then proceed to present it in an organized fashion so that this scientific question is addressed in the most effective way and in the most enjoyable for the reader way. This means that sometimes you don't present your data in the order you obtain them, in the order that you perform the experiments, but in the order that makes more sense eventually and in the context of the manuscript. Absolutely critical. Ensure that every claim you make has accurate data to support it. Don't exaggerate and don't underpresent your data. It's very important to be able to appreciate what your data is telling you in a way. What conclusions, safe conclusions, accurate conclusions can you reach from them? Don't generalize unless you have the information to support a more general conclusion. So these are all very important tips in reaching the point of having a structure for your manuscript. And once you reach that point, we're moving into the chapter of how do you communicate your research findings. There's different types of medical research, different types of study, and this is important to keep in mind because based on the type of study that you performed or you want to perform, you will need to choose the appropriate type of manuscript. So we have primary and secondary types of medical research. The primary one includes preclinical, clinical, and epidemiological studies. The preclinical can be divided in theoretical and applied. The clinical and the epidemiological can each be divided into observational and interventional. As you can appreciate, each different type of study will have both different requirements in its implementation, but will also give you different types of data that will need to be presented in an appropriate way and eventually published in a suitable journal for this type of research. Then we have the secondary types of medical research, and by that we're referring to meta-analysis and literature reviews. Now, there are different types of journal manuscripts. There are the original research articles where your research observations will be published. There are case reports, a different style, a different type of manuscript that we will be discussing in due course over this presentation. These are usually the most common and simple in a way if you want types of manuscript. Then we have the more common, more complex types of manuscripts, and these are for the publication of clinical trials data, systematic reviews, meta-analysis. Then we have some uncommon, if you want, and simpler than the middle group types of manuscripts, and these involve the brief communications, letters, frontier articles, technical reports, how do I do it type of manuscripts, commentaries, or opinions. So these tend to be much more much smaller usually in size, at least some of them. As I said, they're simpler to put together than the middle set of manuscripts. And they have also their specific aspect characteristics to bear in mind if you are to proceed in the preparation of such a manuscript. So once you've decided and you're clear as to what study you've performed and what type of manuscript you want to publish, there comes the time to start putting your manuscript together. So let's look at the overview of the process, and then we will go into each one of these steps in more detail. So once you've identified the right article type, if it will be a full research manuscript, if it will be a case report, how do I do it, a brief communication, once you've decided on that, you need to go on and assess the coherency of your hypothesis and evidence and decide how you will be presenting it. You need to be careful of the language and the style and try to harmonize this with the requirements of the type of manuscript and the journal you will be aiming at. Formatting and referencing. Keep that in full agreement with the requirements of the journal you will be submitting to. Data presentation is a very important aspect. Figures, tables, we will discuss this in more detail. Supplementary information. This could be a useful chapter when it comes to your manuscript, as often we have more data than can fit in the main part of the manuscript. So keeping the option of supplementary information open and knowing how to use that is important. And of course, needless to say, but I will be emphasizing at multiple points through this presentation, make absolutely sure you avoid any research misconduct. This is disastrous, not only for a publication, but for someone's career. So when it comes to choosing the right type of article, keep in mind that if you will be putting together clinical trial type of manuscript, systematic review, or meta-analysis, this requires extremely strong evidence. So any of these three manuscripts requires and makes available to the reader extremely strong evidence. Strong evidence also need to be available for research articles, original articles, brief communications, letters to the editor, frontier articles, and technical reports. Somewhat weaker evidence, but accurate nevertheless, go into case reports, how do I do it, commentaries, and opinions. Let's look at an example. You want to publish a unique and striking clinical outcome of a disease. What type of article can you publish based on it? Which journal should you aim at? These are the first questions to answer once you have all your data together and you're clear as to what your observations and conclusions are. Then you can decide on the type of article and you can decide on the journal that you will be aiming at. Once you've decided on that and you've answered that, then you need to start putting this information together in an appropriate fashion. And the type of manuscript, in a way, affects how you will prepare the outline and build the content. Your article will need to have, once completed, a title, an abstract, an introduction, methodology, results, discussion, conclusions, bibliography. This is the IMRAD format, if you want, with IMRAD coming from the first letter of each of the main parts of the manuscript. Although this is the typical structure of a research manuscript, the order that we go about putting these sections together is usually different. The recommended order for writing your manuscript is starting from the methodology, which is the simplest and most straightforward part because you know very well exactly what you've done. So you describe carefully the methodology, then you present the results, you put together the results, and this includes the text, the figures, the tables, any graphs you want to make, any schematics, and so on. Then we usually go on to write the introduction. And yes, introduction comes third and not first in the writing order, because once you've written your results, you usually have a much better feel for how your introduction should best be structured and what information it should contain in order to best support and best prepare the reader for what will be presented in the results section. We will talk a lot more about that in the slides to come. After the introduction, it's recommended to write the discussion, where you discuss your findings, their significance, and we will see exactly what else you can discuss in the slides to come. The last part in the main text of the body is the conclusion. What are the conclusions from this work and from this manuscript? Last, you write the abstract and you choose the title of the manuscript. So let's look at each one of these points in more detail because there's really a lot to be said about how to best prepare each one of those sections. First of all, we will be writing the methodology. The methodology must include details of animal models or biological material. It should include the full details of which animal model was used, for example, at what age, which gender, under what conditions was it bred, if it's biological material, where did it come from? Did it come from humans? Did it come from animals? How was it received? How was it kept? Under what conditions? How was it processed? How long has it been in these conditions? For example, it's been in liquid nitrogen for 5 years. The age of the individuals or the animals, where it was obtained from, and so on and so forth. You need to be as detailed as necessary so that anyone who reads your method section will be able to replicate exactly what you did. So full details need to be available. Very important that you specify that the necessary ethical guidelines were followed, whether it was animal models, human biological material, whatever ethical guidelines are suitable for the type of work you've been doing, you need to specify them and you need to clearly indicate that you've been following them. Additionally, you need to indicate that you have received ethical approval by the suitable committees in your institute or your country for this type of work. So you need to have these ethical approvals, and this needs to be mentioned in your methodology. The methodology will also include the experimental or sampling design. Which samples were used, how many samples were used, for example, what different time points were used in your study, what times of day or night, if this is relevant, etc. The actual experimental design is extremely important. And in some cases, when the experimental design is more complex than usual, sometimes scientists choose to present it in the form of a diagram. And this is often the case in clinical trials or clinical studies where you need to clearly present the different groups and the different conditions that were assessed in the project. You also need to clearly mention what reagents you used, including the brands. which products were used for this project. Needless to say that not all reagents, if they come from different companies, perform the same way. So if you're using a specific antibody, you need to indicate which company you got it from, because the same antibody from a different company may perform in a different way. So if someone wants to be able to repeat your experiments and get the same result, then you need to be very clear as to what it is that you used. Concentrations. In what concentrations did you use the different reagents? Incubation times, temperatures, and so on and so forth. There's really a lot of parameters, but you know best what parameters you had to use in order to perform your study, and in order for someone else to be able to replicate this work. I know I'm repeating some concepts over and over. That's because I believe they're extremely important, and they're really at the heart of putting together your methodology in this specific situation. You also need to talk about any advanced equipment, software, and/or settings that were used. Settings in your software. If you're performing, for example, a bioinformatical analysis and someone wants to perform the same bioinformatical analysis, you need to tell them exactly which settings you used to get the results you got. So keep all this in mind. The data analysis aspect also needs to be carefully described. So statistical analysis, what tools did you use, which methods did you use, which thresholds did you use? All very important. Don't forget to include, both also under methodology, study location, the time of data collection. And this is about clinical manuscripts, right? So for the clinical work, you need to bear in mind some additional parameters. Again, experimental or sampling design, inclusion and exclusion criteria, clinical demographics. Again, we have data analysis, including statistical analysis, and here emphasizing even more so ethical guidelines. So you'll see how between basic and clinical, basic research and clinical type manuscripts, there are largely overlapping concepts, but there are some specific aspects that you need to consider additionally to the more traditional aspects of a manuscript. Now moving to the results section, which is the second part that we recommend one writes. The results must include distinct sections for each subset of experiments that were performed, for each subset of findings that will be presented. And you need to be clear in your mind what these different sections will be before you start writing them down, so that there's no overlap in the information that you will be presenting, and so that each section is independent, complete, and coherent. These sections need to tie well one after the other, and there needs to be a rationale as to how you transition from one to the other. In each section, be clear about the aim of this subset of experiments. Be clear as to the rationale of the strategy. Describe how the methodology was applied without repeating what you did, but clearly say how you applied the methods that were indicated. So which set of methods did you use for this specific subsection, each specific subsection of your results? And then present, describe in a very detailed and objective manner the observations of your study. So extremely important. You need to be very detailed and you need to be very objective. In the results section, we tend to use figures and tables. There's different occasions, there's different data for which either the figures or the tables may be better suited. You need to decide, depending on the data of each set of experiment, what the most appropriate format of presentation would be. For example, if you've used microscopy, it's usually best to present photos of what you saw under the microscope rather than data in a tabular format. However, on other occasions, if it is multiple measurements that you need to show, maybe a table or a graph would be better suited. We will talk a little more about that in the slides to come. Independently of whether it is a figure or a table or a schematic, it needs to be very high quality, the photos need to be a very high resolution, and sometimes the journals specify exactly how high resolution they require. Absolutely critical that there's no manipulation of photos that changes the observation and the conclusion that is reached from it. Figures and tables need to have the appropriate size so that they're clearly visible, but also fit into the pages of the journal. The choice of colors needs to be appropriate so that it's aesthetically pleasing. Before that, informative, but also in line with any requirements the journal may have if they do. Diagrams, bear in mind, can also be very helpful, so it's another tool that you have available and you're welcome to use depending on what it is you will be presenting. Supplementary material is optional if needed. If your results don't fit or some of your figures or tables don't fit in the main manuscript, or if they're too large or too many or too detailed, they can be available to the readers as supplementary material. Another important point is consider depositing your raw data in public databases. In some circumstances, such as in the case of microarray or next generation sequencing or proteomics data, this is actually a requirement by many journals. It's not just an option. Choosing the best format. For your results, it's important to decide whether you will be using text, tables, or figures to present it in the most clear and easy to communicate to the audience fashion. So text is great for qualitative data with simple relationships, for quantitative data, only for important or highly significant findings, or is good for presenting peripheral or supporting data. Now, tables are great for presenting precise numerical data for many variables if you have many measurements. It's useful for comparing contrasting values, for demonstrating presence or absence of specific data characteristics, and for data then can be presented through a table in a more concise, reader-friendly fashion compared to text. And then you have the option of figures, which is what readers usually enjoy the most. Figures are used to present photos of results, to have a visually appealing, aesthetically pleasing representation of the data, to summarize research results, to demonstrate patterns, trends, or relationships between data sets. So let's look at some examples. And here we have an effective textual presentation example. So on your left, you have a set of values of age and gender of 50 patients. In the table at the top, you have the demographics of the patient population. So you see how this set of data could be organized in a tabular format. So you have the numbers of subjects, you have the age of the different subjects. If you were to describe this as text, you have two different versions of it here, and I will not be reading through the details of the first, which would be the inappropriate one. Our population n equals 50 included 23 men and 27 women. This would be a more tiring and inappropriate way to present it, whereas a more appropriate would be the second box, where you have a description that goes like this: the mean, parenthesis, plus or minus standard deviation, age of the overall population, and 50 was 38.36 plus minus 15.38. You provide the range. You indicate that these are years. Then you talk about men and their demographic specifics, where significantly you indicate by how much younger than women, and you indicate their specifics. So through this presentation, you make clear to the reader what the facts are and what the main observation here was. You don't leave them to try and reach conclusions by themselves. You prepare and present the information in the most informative and easy to appreciate fashion. Here are some tips for preparing a table. Now, first of all, ensure a clear, to the point, brief title. So each table needs to have a title and a table legend. The title and the table legend always go to the top of the table, as opposed to figure numbers and figure legends, which go to the bottom of the figure under the figure. So for the tables, the title and the legend go to the top. The table legend needs to be brief, clear, and comprehensive. All the key points of the table information need to be indicated in the table legend. If necessary, you can include bibliography either in the table itself or as a footnote or at the end of the table legend. It's up to you and also depending on the journal requirements. If footnotes are needed, you can always place them at the bottom of the table. Footnotes can be used for clarification of specific points or additional information that you may want to make available for specific boxes in your table. The table needs to be clear, as I said, well organized, and the take-home message needs to be evident as easily as possible. Usually we don't use color in the tables for publications, but if color can help the take-home message become more evident, you can consider using it. Just a small parenthesis, bear in mind that journals sometimes charge for color material, so this is one of the reasons why tables are not usually in color unless absolutely necessary. Let's look at some more examples here. You can see how the table number goes at the top, followed by the table title with all the important information. This table is here to show the comparison of what? Of fold change. Fold change of what? Of mRNA expression when? At 24 hours after baseline. And how many samples are included in here? N equals 12. And then in your table, you have the different columns and different type of information. And at the bottom, you have a footnote with the abbreviations. What do the different abbreviations mean? And the reason for that is that you don't want to put the full name for each molecule in the table. It would make it too crowded, too busy. To create your table, you can use the table function in Word, or you can use Excel if needed. Representative titles with overall sample size, column and row headings have units and sample size. The primary comparisons are horizontally. There's cause and effect and before-after relationships, left to right, and then top to bottom. So you can see how there's a clear structure that one needs to decide upon and use when preparing a table. The order of the footnotes needs to be logical. So here, for example, we're providing abbreviations and they are in the same order as the order that are presented in the table. Consistent statistical parameters, you need to show consistent information across columns or rows, and the size needs to be manageable. If you're dealing with a table that has hundreds of rows or hundreds of columns, then this, of course, could not be included in the main part of a manuscript would have to be supplementary if it's absolutely necessary to include such a big table. Okay, so what about the diagrammatic presentation? Diagrammatic presentations are useful to highlight patterns or trends in data, to demonstrate changes over time, to display complex relationships among quantitative variables, to clarify and explain methods, to provide information that will supplement complex concepts, to illustrate pathways, pathophysiology, and mechanisms, to present conclusions of findings or hypotheses based on findings. For all those reasons, a diagram would be a great choice. Now, if tables or diagrams cannot be the best way to present your data, then a figure may be. So when we're preparing a figure, here are some tips to bear in mind. First of all, be careful of the image resolution, and this is step number one and tip number one, because this is the first thing we do when we capture, for example, a photograph that we want to turn into a figure. So keep in mind throughout your project, I would recommend that the photographs you take of different observations, of different results, of different experiments, they are at a sufficiently high resolution, because you can't afford at the end of your 1, 2, 3, 5-year project realize that the images you took months or years ago were of a poor resolution and you need to repeat your experiments in order just to get better pictures. Make sure high quality is ensured and high resolution is ensured right from the start. Tip number 2, take care into the file type. Do you prefer, and does the journal prefer, TIFF, BMP, JPEG files, or something else? Make sure you prepare your figures accordingly. What is the optimal size and shape? Ensure you have clear image labels and units. Be aware of the different figure guidelines that the journal you will be submitting to has, and ensure that you've implemented those guidelines. You don't want to have tabular data in a figure. Be aware of the color format that is recommended by the journal, if there is a recommendation, and ensure you implement that in the presentation of your figure as well. Some additional tips. When it comes to preparing graphs, make sure that they're free of irrelevant or extraneous marks or text. All lines and graph points must be distinct with considerable contrast. Ensure there are no typographical errors. Ensure that acronyms, abbreviations, and symbols used in figures are consistent with those in the text, and that's a common mistake. We often make this mistake when we try to fit things into a figure because space doesn't seem to be sufficient. So we may choose to change the abbreviations or change acronyms or change symbols, but no, this is not good practice. To facilitate and make the reading more clear and enjoyable for your reader, make sure that everything is consistent between text and figures, between tables and figures. Maximum consistency is required. Maintain uniformity in style, for example, font, letters, cases, and size of figures. Figures can be provided within the text, close to where they cited, or at the end of the manuscript. I would advise, though, keeping them at the end of the manuscript, because this is what most journals prefer. And then when they do the final editing, they tend to take the figure and place it as close to where it's cited in the text as possible. So yeah, the option is open, but putting them at the end is preferable, so after the bibliography. Resources now for making charts. There's many different tools available out there, and these are just some examples to help you get started. Examples of software that you can use to make charts for your manuscript: GraphPad, Prism, Sigma Plot, SPSS, MATLAB, etc. Once we've gone through our results, we need to put together an introduction. The introduction explains why your research study matters, and also provides the basics so that the reader will be able to appreciate where the field stands, why your study was important and needed, and why your findings are making a novel and significant contribution to the field. So you need to provide appropriate background of the topic. What's the bigger problem being addressed here? Why is it so important that you chose to devote time and effort in addressing it? What's the information available to date? What are the major open questions in the field? What are your specific aims and hypotheses in this study? And, of course, provide appropriate citations throughout to support every single piece of information that you offer. At the end of your introduction, the reader must be able to appreciate why it's worth reading your paper, and why what you've been doing was needed in the field that you worked in. Now, after we write the introduction, then we go on to write the discussion. So now you've already provided the reader, or at least you have prepared, not provided the reader, but you have prepared the introduction, the methodology, and the results. Now comes the discussion section, where all this information that you've put together so far needs to be tied well together, needs to encapsulate the study design and main outcomes in a paragraph, and that's usually the first paragraph of the discussion. And then you go on through distinct individual paragraphs where each major outcome is discussed. And when we say discussed, this means discussing your findings, what they mean in relation to related studies in the field. Are they supporting other findings, other hypotheses? Are they disputing them? Discuss unexpected findings. Discuss the significance of your observations and where is this information leading us in terms of answering the biological question, the scientific question at hand. Provide logical reasoning for any discrepancies. Try to be as objective as possible. If there are limitations in your study, and there always are limitations in studies, it's useful to be clear and direct about them. So you may want to refer to issues related to the study design, study population, sample size, study duration, the factors that were evaluated, the methodology that was chosen, the number of replicates, the material used, etc. There may be limitations in your study in any of these parameters, and for a good reason too. It's often the practice to present these limitations along with the explanation why you couldn't have done it in any other way. And this was really the best possible or the only possible way forward. Acknowledging this limitation does not take away from your study. It just puts the findings of your study in the right context and demonstrates that you've taken into consideration these limitations when it came to interpreting your findings. After the limitations, if you include limitations, and at the end of the main text of your manuscript come the conclusions. So the conclusions should summarize the key findings and highlight their short, medium, and long-term implications in their respective medical and scientific field. The conclusions are usually a much smaller section than all the others, but nevertheless very important because this is the take-home message. At the end, you put together the abstract and the title of your manuscript. The abstract must convince the reader to keep reading. So you want it to be clear, you want it to be informative, reader-friendly, but also motivational in encouraging the reader to read on. You want to include key points from the background, the rationale, methodology, results, and conclusions. You want it to be a self-sufficient miniature article, if you want. That's actually the best way to describe it. You want to fit a miniature article in the 300-word limit or whatever word limit the journal you'll be submitting has. Provide sufficient detail to ensure inclusion in meta-analysis and reviews, because often these future studies will be mining data from your abstract to decide if they are to include it in their meta-analysis or reviews. But very importantly, make it appealing, highly scientific, highly objective, highly clear, lay reader friendly, an abstract that will make the reader want to keep reading. And there comes the title, the reader's first impression of a research paper. From your experience, you probably already know that when we go searching through PubMed to identify manuscripts or papers, publications to read, we first look at the title. It's the title that determines whether we will open an article and read it. A title must be concise, accurate, original, captivating, use field-specific keywords, and explicitly state the most important findings. Although it's the smallest piece of text in a manuscript, it's just a sentence or less than a sentence, it's actually just a few words, the title has an extremely important role in a manuscript. So choose your words very carefully when it comes to putting together the title. Citations. An in-text citation needs to be inserted in the manuscript wherever a work has been influenced by someone else's work. Citations is a whole big chapter in its own. Authors must provide citations while directly quoting someone else's work, paraphrasing someone else's work, presenting results on the basis of an already published work, and you also need to decide how you will be formatting your references, and this is dictated usually by the requirements of the journal you will be submitting your manuscript to. The medical literature generally follows either the Vancouver style or the Harvard style, citation styles, but regardless what may be more common, you definitely need to be aware of what the specific journal requires and format your bibliography accordingly. So bibliography is really the list of the sources that the author has referred to while conducting their research. Always check the journal's recommendations, and the information you will need to have there, as you probably already know, is the author, publisher name, editor name, the title of the research article, book, or web page, the date of publication, the year of publication, the place of publication, a web address. You need to indicate page numbers when we're talking about text and not web pages, volume number, etc. For example, this is what a Vancouver style citation would look like, where we first have the author names, but you can only give so many author names. You can give 6, and after that you use et al, so you don't provide all the author names. Then you have the title, you have the title of the journal, you have the year of publication, volume, issue, page number, and the DOI, the date of issue number, which is the unique identifier of the article. There's many different tools for managing your citations and references, and they can be extremely helpful. Some examples of commercial reference management software packages would be EndNote and RefWorks, but there are also free tools such as Zotero, Mendeley, Citavi, and others. It really does make life much easier to use a reference management tool, and I would definitely not recommend doing it manually in a manuscript. The quality and the accuracy is highly likely to have flaws compared to if it's done automatically. Now let's go through some key points for medical publication and research ethics. Fundamental principles of research ethics that need to be followed throughout a research project: honesty, objectivity, integrity, caution, responsible mentoring, respect for intellectual property, confidentiality. Important points are the openness, the respect for colleagues, social responsibility, non-discrimination, competence, legality, animal care, human subject protection, and responsible publication. I know I've just bombarded you with a large number of very important points, and research ethics is a whole big chapter in its own. But I've tried to summarize here the key points because they need to be taken carefully into consideration in any research project right from the start, so that when it comes to publishing your work, you do not encounter problems. Medical ethics, again, you need to ensure from the start of your project if and where consent is needed. You need to obtain that and have this available wherever needed from the start of your project, so that you don't reach the end of a project after many years, possibly, where you're asked to show informed consents and you don't have them. Informed, of course, means that the patient or his/her attendant understands the mode of treatment or procedure or the reason for obtaining a sample, knows how the sample will be treated, stored, etc., expresses authorization, permission, a choice by patients for the doctor to act in a particular way. It clearly indicates that it's voluntary and that they have the right to withdraw their consent. And also, during the process of informed consent, if invasive tests or treatments with significant risks will be implemented, this needs to be carefully written in the consent, but also carefully explained to the individuals. There's many resources for information and ethics, and I would like to point you to the World Association of Medical Editors, as well as resources such as co-publication ethics flowcharts, equators, reporting guidelines, ICMJE, and IMRAD. These are resources where you could find a lot for more information. There's also important ethical points that need to be taken into consideration in clinical trials, and I'm just laying out some of the key points, by no means an exhaustive list. It's very important to adhere to good clinical practice, an international standard. Compliance with GCP ensures credible data reporting, ensures the safety, confidentiality, and health of all subjects. And actually all NIH defined or NIH funded clinical trials are required to follow GCP. Also, GCP training should be renewed every 3 years. So it's something very specific, very clear rules, very clear guidelines. The ICMG has a clinical trial registration policy. I won't take you through all the details, but I would encourage you, if you're involved in clinical trials, to go through the ICMJE's information and be aware of all the key points that relate to clinical trials, so that you can implement them in your study in a timely fashion, in an appropriate fashion. Very important to be aware of publication ethics. These include transparency of the funding for your research or publication, details of clinical trial registration numbers, any conflicts of interest, all these needs to be disclosed. Authorship and acknowledgements, there's very clear guidelines as to who, how, when should be or should not be an author, should or should not be acknowledged. Registering clinical trials for any of you performing clinical trials is also important, and again should be done through public clinical trial registries. Some additional tips for researchers. Very important points that I'd like to point out in the course of this presentation. Integrity and accountability for the scientific and social impact of the paper is very important to keep in mind and at heart. Follow authorship guidelines and resolve any dispute courteously. People with no significant contribution should refrain from claiming undue credit in a publication. People making significant contribution, on the other hand, should receive their due credit. Crucial part in any research project, in any publication. Avoid research misconduct at all costs. It could really not just ruin your project, your publication, but it could really damage your career. Data fabrication and falsification are absolutely unacceptable. Plagiarism, it's a big no-no. Careers have fallen apart because of that. Inappropriate allocation of credit, it's not right. There could be official complaints for that, and it can lead to problems for your publication even after it's been published. Dual submission and dual publication is unacceptable, not allowed. Only preprints are allowed in terms of prior submissions. Citation manipulation is not acceptable. Copyright infringement, be very careful. Sometimes copyright infringement may take place accidentally. Make sure you know the copyrights of any material that you want to reuse. Duties of authors: to maintain transparency, to declare any competing interest, to strictly follow authorship guidelines. You need to maintain originality and acknowledge all sources. Provide access to data and retain it for future reference. Notify editors of any fundamental errors that you may detect post-publication. This is very important, and unfortunately there are cases where retractions have taken place. So although it's extremely important to make sure everything is in place, everything is accurate, true, and appropriately presented, if you discover that something went wrong even after publication, do notify the editors and try to find together with them the best way to resolve the problems. Of course, avoid duplicate publications, comply with relevant human and animal rights laws and guidelines, and maintain reporting standards for data and image integrity. These are different points for different areas, but all very important, and they are responsibilities that you have as an author. Now, one small chapter I would like to touch on for those of you who are interested in putting together case reports because you're working in the medical field. Here are some additional tips and advice to bear in mind. So what are case reports? Case reports serve the purpose of providing essential sources of information for better care of patients, as they enlist important scientific observations that are not evident during clinical trials. The three primary functions that case reports serve are to identify new areas of research for verification in subsequent observational studies and clinical trials, documentation of rare or unusual observations, and clinical material that they provide to medical professionals or researchers to revisit noteworthy lessons. What qualifies for publication as a case report are previously unreported observations of a known disease, use of unique diagnostic procedure or procedures to diagnose a disease, previously unreported complications resulting from a procedure. So any of these could really stand as a case report. A case report will include an abstract, keywords, introduction, how did the patient present, diagnostic focus and assessment, therapeutic focus and assessment, what therapies were tried, what worked, what didn't, follow-up and outcome, a discussion, a conclusion, and then a patient perspective and information on patient consent. So slightly different structure from what the typical manuscripts we've been talking about have. And in this slide, you can see the actual questions that we try to answer in each section of a case report. Again, this doesn't have to be exactly this way in every case report. It will need to be adjusted based on the case that you are presenting and the parameters that you're presenting. A typical case report checklist would be: does the diagnosis satisfy accepted criteria? Is the length of the case report less than the maximum allowed by the journal? Do all authors meet the ICMJE criteria for authorship? Does the report appropriately describe the patient's condition? Is the case distinguishable from existing literature? Is there unwarranted extrapolation and generalization? And of course, one needs to be very careful of that, especially in case reports where we're basically working with a minimum number of samples. Are citations obscure, outdated, or inaccessible sources? You need to be careful in the citations that you use. Make sure that they're up to date and appropriate. When it comes to evaluating a case report, here's the Pearson's conceptual scheme for quality. Evaluate the documentation available, the uniqueness, the objectivity, the way the data is interpreted, and the educational value. Now try to score each one of those points and decide if your total score is high enough to merit publication of this case report. With this in mind, we're coming to the end of the presentation. I would like to thank you very much for your attention. I hope you found useful information that will help you in the course of your manuscript preparation and your research project. And I would like to wish you best of luck with your publications.
Thank you, Dr. Despina. Our attendees have certainly gained a lot of critical information from this session. We would once again like to thank you all for joining the webinar. We would request you to please fill the feedback survey displayed after you leave the webinar. Your participation will allow us to evaluate the effectiveness of our webinar. Have a good evening.